Now that we have identified DNA as the molecule of heredity and looked at its replication of how you get DNA into the next generation, the next question is, well, how do we actually use DNA? How does the cell do something with DNA in order to make something happen? And the big answer to that question is, well, the cell is going to use the DNA in order to make proteins, because proteins are going to be enzymes, and enzymes are going to control every process that goes on in a cell. So really what we need to take a look at next is, how does the cell use DNA in order to make proteins? So let's call that process protein synthesis. There are two big parts to protein synthesis, and these two big parts have become known in biology as the central dogma. This is kind of the basis of how DNA is actually working. And the two parts are called transcription and translation. So you see here in this diagram, this is what's going on in a bacterial cell, and this is what's going on in a eukaryotic cell. Both cells have transcription and translation. They happen in slightly different places because a bacterial cell doesn't have a nucleus. Transcription and translation both happen in just the cytoplasm. In a eukaryotic cell, transcription is going to happen within the nucleus, and translation is going to happen outside the nucleus. And so we have to have a way of getting that information to the outside of the cell. Think of DNA as a very valuable material. The cell doesn't want to damage its DNA. So as an analogy, think about a library, and it has a very rare book where handling that rare book could easily damage it. The paper pages are old, they're going to crumple up. So when researchers want to use that rare old book, what do? They make a copy of it, or the librarian's going to make a copy of it. That's what your cells do with the DNA. DNA is so vital that in order to work with the information that's there, the cell is first going to make a copy of it. The copy is called messenger RNA. Messenger RNA. The process by which the cell makes messenger RNA is called transcription. Transcription is how we go from DNA to RNA. It's the same language. It's both nucleic acids. RNA and DNA are very similar. And then we need to go from the language of nucleic acids, DNA and RNA, into the language of proteins, into the language of a sequence of amino acids. So we have to translate from the language of nucleic acids to the language of proteins. And that's why the second step in protein synthesis is called translation. So here we see we're going to have our DNA, we're going to make a copy in messenger RNA, and then that's going to go in a eukaryotic cell out to the cytoplasm, where it's going to get used at the ribosome to make the protein. So before we start, let's make some comparisons between RNA and DNA. We've alluded to a couple of things. We haven't really said very much about RNA. We know a lot about DNA. We know DNA has a sugar phosphate backbone, and so does RNA. The one thing that's different in the backbone is that the sugar is not deoxyribose. In RNA, the sugar is simply ribose. Now, when I say simply ribose, it has one more oxygen than the sugar that's in DNA. Not particularly of major consequence for any functional attributes that these two molecules have. Second thing is that you'll notice DNA is a double-stranded molecule. RNA, almost always, there are some exceptions, almost always is single-stranded. In other words, there is no complementary side to that DNA molecule. And the third difference is that it has one different nitrogen base. It has guanine, it has adenine, it has cytosine, but the thymine has been replaced with uracil. So when we encounter a need for base pairing and we've got an A in RNA, the U will pair with A instead of a T. So we still have C and G, but now we're going to have A and U in RNA. So those are the three differences. Sugar is ribose, it's single-stranded, and it uses U instead of T. So this is going to be, I'm going to try to show this without any sound because I want you to just have an idea of where we're headed with transcription. So let's see if I can make that happen. I don't want to worry about any of the details. Interesting. Okay, let's try.
Transcription is the first step in the expression of a gene. It is the formation of a specific RNA sequence from a...
Okay, so what we're going to see here is that there's a region on DNA called a promoter. And at that promoter, that's where the enzyme RNA polymerase is going to begin. So it is super versatile. And that means that it can do a lot of the things that in DNA we needed multiple enzymes to do. And then along one side of the DNA molecule, we're going to add RNA nucleotides. So you can see them joining until it reaches kind of a termination sequence. It's going to keep on building. It's going to be doing its base pairing. It's going to be pairing A and U and C and G and A and, well, sorry, the T in the DNA is going to be paired with an A in the RNA. And then we'd have G and C, and anytime we run into an A, we're going to pair it with a U. So we do have base pairing between the DNA and the RNA. And that's going to keep on continuing until that RNA polymerase, which is kind of that green blob in our picture here, runs into a termination site. So in a moment, you'll see it hits the termination site, and now that RNA is going to actually come off and be available for use. So big picture of what's happening here. In order to make messenger RNA, that RNA needs to unwind. And RNA polymerase, the enzyme, can make it unwind. And then you have two sides to the DNA molecule. Well, just like in DNA replication, we can only build, RNA polymerase can only build new RNA, the new strand that we're working with, in the 5-3 direction, so that the new nucleotides will get added to the 3' end of the growing RNA strand. But RNase can unwind it, it can unzip it, it can hold it open, and then it will continuously make RNA until it hits that termination sequence. And so the side that it uses is going to be known as the template strand, because that's the side that is the template for this RNA polymerase. And then the other side is actually going to become known as the coding strand, because if you start to look at the RNA that gets made and this, what's called the coding strand, or often the sense strand, so you'll see that the template equals the nonsense strand and the sense strand equals the coding strand. What's happening here is this new RNA that gets made is identical to the side of the DNA molecule that's not being used as the template. So look right here, RNA has a U, well, there's a T in DNA, then there's an A in DNA and an A in RNA. There's a C in the DNA and a C in the RNA. Wherever there's a T in the DNA, there'll be a U in the RNA, but there's a G and a G and a C and a C and a C and a C. So RNA and the sense or coding strand are identical. The template strand or the antisense strand is the opposite. It is the base paired side of things. So if we were to sketch how transcription occurs, we would do something like this. We'd start out with a DNA molecule, and then that's going to be opened up very much like a replication fork. Oops. A replication fork. And then along one side, so let's say we would want to have, we want to have the 3' there, so we're going to have our 5. So this would be the 3' side. RNA polymerase, so we'll represent RNA polymerase like that, is going to start to add new RNA nucleotides at 5 to 3 end. And RNA polymerase is just going to keep on going down the strand until it hits, let's say we'll call that the termination sequence of the DNA molecule. So there's a particular region of the DNA called the promoter. Make the promoter right in here. The promoter is the very beginning part of the gene that needs to get copied. So think of the promoter as the start spot for the RNA polymerase. So the RNA polymerase will come in, it will bind here at the promoter, it will start to add new nucleotides, new RNA nucleotides. So this side is the RNA that's forming according to the base pair rules. It will make the new RNA along the template strand. The opposite side would be the coding strand of the DNA. So here our DNA is in the process of unwinding, and that's what the DNA polymerase can do. Here, sorry, well, yes, it's unwinding. And as our RNA polymerase moves down, what we're going to see is that the RNA is actually going to come off. So let's say a moment later, if I were to draw this same picture, we would have now something like this. Where our RNA has started coming off. There we go, it started coming off. And eventually it's going to hit that termination sequence. And now it's done. It's finished in terms of copying the DNA. And our DNA, so the next moment later, will have rewound because the RNA polymerase will have come off. This is our RNA polymerase. And now we have this piece of RNA. At this point, if we are in a prokaryotic cell, this would be finished messenger RNA. If we were in a eukaryotic cell, like your cell, this would be what we would call pre-messenger RNA. It's got to go through some processing. It's got to be changed a little bit before it's going to go out to the cytoplasm where it's going to get used by the ribosome. So let's take a look. Here's a much nicer picture of what we've just been sketching. Here we see our RNA polymerase, which is this large purple circle. It started up here at the promoter region, and it is unwinding the DNA. Along one side, the RNA polymerase is adding RNA nucleotides. The new RNA is growing in the 5 to 3 direction until it comes to that termination sequence, and then the RNA is going to be free to get processed if you're in a eukaryotic cell. And so I think you get the idea, and I'm going to skip all these pictures. All right, so now what kind of processing are we talking about? It turns out that if this was a piece of eukaryotic DNA, so this was your chromosome, and let's say that that was one gene's worth of DNA on your chromosome. It turns out that in that gene, there are sections that are junk. That might surprise you. Within a gene, there are pieces of that gene that we don't want to use. Those pieces are called introns. So that one, and that one. The pieces that we want to use, the pieces that we want to keep, the pieces that we want to express are called exons. So in this gene that I just drew, this would be an exon, this would be an exon, this would be an exon. And we'll talk about why these might exist, but at this point, they do exist. So what has to happen is when this DNA molecule, so there's our DNA molecule, has been used to copy that gene and make this RNA has those introns in it. It has sections that don't need to be there, that we need to get rid of. We need to cut out those sections that we call the introns. And so before the RNA leaves the nucleus, so here would be the nuclear membrane, we want to cut out these introns. There's going to be this processing that happens of the pre-mRNA. And it's a little complicated process. I'll show you a quick little video of how it works. But once you remove the introns, then the cell is going to join this exon to that exon to this next exon. And now that's almost finished, but we're getting closer to the messenger RNA. So what happens, kind of, how does that look? Once you make this messenger RNA and then you cut out these introns, so now you have this section of messenger RNA, there's a little bit more processing that's also going to happen. At one end of the messenger RNA, the cell is going to add a 5' cap. It's actually made out of guanine. So there's going to be this cap, guanine cap that gets added. And the other thing it's going to do is it's going to add a whole bunch of A's, whole bunch of A nucleotides, adenine nucleotides out here. So we've got this piece of RNA, the introns are gone. We put a G here, we've got all of our RNA, and then we put a whole bunch of A's out here. The purpose of adding this cap and what we call this poly A tail, it helps prevent the degradation of the DNA out in the cytoplasm. So it's going to hang around longer when it's got this cap and that poly A tail. So how does the cutting process actually work? Little kind of details here. Basically what's going to happen is there's a set of proteins. They're called SNRPs, kind of a fun name. And there's usually 4 of them. 4 of these SNRPs are going to come together at the two ends of those introns on the pre-RNA. And they're going to kind of bring them together. As you can see, they're forming this loop. And once they're together, they're called a spliceosome. They're going to join the 2 red exons and cut out that kind of coral colored intron. So here's how it works.
Most eukaryotic genes are composed of numerous short coding sequences called exons, embedded within stretches of non-coding sequences called introns. The initial messenger RNA molecule, or primary transcript copied from a gene by RNA polymerase, is a faithful copy of the entire gene, including introns as well as exons. Before the primary transcript is translated, the introns are removed by a process called RNA processing or RNA splicing. Particles composed of proteins and a special type of RNA called small nuclear RNA, or snRNA, play a role in RNA splicing. One kind of small nuclear ribonucleoprotein, snRNP, contains snRNA that can bind to the 5' end of an intron by forming base pairs with complementary sequences on the intron. A different snRNP binds to the 3' end of the intron. Additional introns interact, causing the intron to form a loop, thereby bringing the two ends of the intron together. The large complex of snRNPs, called a spliceosome, then excises the intron and the exons are joined together. The snRNPs are then released.
Pretty snazzy. So that's how we're going to process our pre-messenger RNA. And once that's done and we've added the cap and the tail, now we finally have mature messenger RNA, only composed of exons, that can now leave the nucleus and go out to the cytoplasm. The last thing I want to mention is that it turns out that when these exons get joined together, that different exons can be joined with other exons. Let me say that another way. So here we have our DNA, and you can see where the exons are located. When the cell splices or joins those exons together, sometimes it might join 1 and 4 together, or it might join 2 and 3 together, or it might join 1 and 3 and 4 together, or it might join 2 and 3 and 4 together. This is called alternative splicing, so that from one gene, from one segment, you can actually get a whole variety of different proteins, which is one of the reasons why we actually have a few number of genes, like making up a person, somewhere on the order of 20 to 25,000 genes, because we can make a host of different proteins from any one gene. And we'll talk about that some more, and we can relate that to the immune system as well. And that's what I wanted you to understand right now. So we'll move on to translation the next time we have some time to talk about how DNA works. Thanks.